Great, thank you very much, and thank you all for hanging on for late in the afternoon for yet another talk. So yes, I'd like to talk about the use of a novel phenotyping strategy that we've used to select for cereal root traits. So what I'm going to talk about is firstly the target environment, because the traits that you need to select for to get advantage in your environment are obviously very much dependent on the environment itself. Then I'll talk about a nice multiple reference parent nested association mapping population that's been developed to incorporate such traits into adapted varieties for our environment, and the high throughput screening method that we've used, and show you that Dr. Cecile Richard was able to use the method to select for root angle in this particular case. And if there's time at the end of the talk, I'll also talk very briefly about some work we've done on developmental genes. So the environments in Australia, we have 3 mega cropping environments. I'm here in the eastern mega cropping environment, where we have summer dominant rainfall. There's the south and the west cropping environments in Australia, where they have winter dominant rainfall, so they're quite different. This little graph here, this little map there, just shows you that we're at a much closer latitude to the equator than large parts of Europe that you guys will probably be familiar with. So if we think about this eastern environment in more detail, it's like a lot of Mediterranean environments in that we do grow spring habit wheat that's sown in the autumn, grown through the winter and harvested in the late spring and summer. But as I say, because it's summer dominant rainfall, that means the crop's actually growing through the winter when there's very little rain. And the reason we can do that is because we've got these deep cracking clay soils that hold a lot of water from the previous summer. So what's going on is that the crop roots are sucking the water out of that profile, and they have to keep sucking deeper and deeper to keep getting more water because of the terminal drought stress that occurs in most years. So that determines the type of root system that we need in our environment. Now, we've looked at a few varieties that are particularly well adapted to our environment and found some favourable traits that are quite instructive to us. We started out by growing them in these large root chambers. They're 2.4 metres wide, 1.2 metres deep, and we can grow plants right through to maturity in these chambers. There's a cartoon here of the side of one of these chambers, which is a glass side where we can do scans of the images. This shows you where the single plant was planted in these chambers, grown right through to maturity, and it shows you the maximum extent of the root systems of a barley genotype, of a standard senescent genotype of wheat in our environment, and a stay green drought resistant genotype in our environment. First thing you notice is that the drought resistant variety has a narrower lateral root distribution than the standard senescent type. And there are some more subtle differences. If we scan the roots at the surface here, we've got the senescent genotype here and the drought-resistant genotype. They've actually got very similar root length density in these shallow depths. But you can see that the senescent genotype's got slightly clumped roots and is leaving some areas less well explored than others, and it's actually not able to extract quite as much water from that layer as the drought-resistant genotype, even though it has similar root length density. If we look down deep at maturity, we can see that there's quite a big difference between the senescent and the stay green genotype, and the stay green genotype has a lot more roots at maturity deep in the soil, which is what we need to extract that soil moisture. Now, there's not a lot of difference in the root length of these genotypes nor the root biomass of these genotypes. The difference is where the roots are, and the difference occurs after anthesis. So what's going on is this genotype's not putting roots out to the side after anthesis, it's growing genotypes deep in the soil, whereas the standard genotype is growing roots out here after anthesis. So it's a matter of the temporal and spatial distribution of the roots that gives the advantage. Now, we can't phenotype hundreds of genotypes in these big root chambers. They take days to set up and months to run the experiments. So, but we do find that in wheat, and it's also been found in barley, that a narrow seminal root angle of the seedlings is correlated with a narrow lateral root distribution of the mature root system. So that's something that we've been able to use to rapidly phenotype large numbers of genotypes. So we've developed a multi-reference nested association mapping population to incorporate some of these traits that we're after into adapted wheat backgrounds. So we've got 3 reference parents here for 3 component NAM populations in this overall population. We've got one for the eastern environment that I just talked about, one for the southern environment, and the western environment that I pointed out at the beginning of the talk. Using the speed breeding technique, Lee Hickey and Cecile Richard were able to develop over 12,000 lines in 1,200 lines in 18 months in 22 different families. Now, Cecile selected one family from each of these populations to subject to phenotypic selection to see if she could move one of the traits of interest, which she chose to be seedling seminal root angle. So Cecile developed this novel phenotyping system with transparent pots. We sow seeds around the edge of these pots so that the roots will grow at the edge of the pot where they can be visualised. They are then photographed, and we can measure root angle after only 5 days, root number after only 10 days at 17 degrees, and we can have 600 plants per square metre, so we can get quite high throughput with this phenotyping system. And Cecile used it to see if she could use actual phenotype to shift the root angle of these plants. So the first thing she did was look at the root angle of the parent plants for the NAM population that I just showed. She did 2 experiments, and this is just the 2 experiments, and you can see that they are correlating well, so the system's working well. But the main interest here is the difference between the parents. So here's the reference parent Suntop for the eastern environment and Mace for the western environment. They've got broad root angle. She chose the family where a Suntop was crossed to Darwar Dry, which has a narrow root angle, and she also chose the Mace family, which was crossed with Drysdale, which has a narrow root angle. She chose also Scout, the parent for the southern region, to be crossed with this SB line. They both have an intermediate root angle, so 2 different contrasting types of crosses amongst the 3. Okay, so she took the back-crossed F1 plants from the NAM families. So those NAM families were made by crossing the donors with the reference parents, then crossing back to the reference parents to give a nice agronomic type. She then let them self to the F2 in the segregating, and she selected those segregating populations to have either wide or narrow tails in the transparent pot system. She then allowed those to self again and selected again in the narrow pots, and only for the Mace Drysdale population she did that for a 3rd generation. And this is the type of result she got. So the first F2 are segregating there, and that's the distribution. So that's the proportion of the genotypes that have specific root angles measured in that pot system. And there's the angle measured for the plants, the narrow parent and the wider parent. So she selected these tails, allowed those to self, and then they were phenotyped in the next generation. So here's the blue wide tail and the pink narrow tail in that first generation of selection. You can see there hasn't been much movement in the phenotype in that first generation. It was the same for the Sun-top Dawa dry population and similar for the Scout SB population. But when she took those and selected them again from the narrow, she selected narrow ones, and from the wide ones, selfed them again and tested the wide and the narrow population again. Okay, she's got a shift of 10 degrees in the mean population mean angle. So that's a good shift, and she carried that down for another generation, only in the Mace and Drysdale population. And when we look at some of those narrow tails in that Mace Drysdale population, we can see that they are definitely considerably narrower than the broad parent Mace, and at least as narrow, and in some cases slightly narrower than the narrow parent Drysdale. So when she looked at the genetics of these tail populations, here in the Mace Drysdale population, you can see we've got the 40 odd genotypes in each tail represented along here, and a number of different genetic loci here. Those with narrow alleles are showed there in pink, or wide in blue. You can see we've got hot spots on 1A, 2B, 3A, 3B, 3D, 5A, 7A and 7D, which have been heavily selected for narrow alleles in the narrow tail and wide alleles in the wide tail. And as we've already known from previous study, it's a complex genetic trait, so you can see those regions just spread throughout the genome. So we've been able to select for seedling seminal root angle. I've talked about its potential utility in water extraction, but root angle can be valuable for other things like lodging resistance and uptake of other nutrients. Seminal root number, which I haven't talked about, is also a trait of interest, can be measured in that system, and we generally find that higher seminal root number is advantageous, 5 roots rather than 3. And but there are many other complex traits that we'd like to select for that are probably going to be quite difficult to do phenotypically, like this root growth after anthesis that I talked about. And there's also the plastic response of roots, which, for example, has been reported by Suzuki Pinto at Matt Reynolds' group, CIMMIT, where the plants grow more roots shallow when there's water available, but more roots deep when there's water restriction. Those things might need marker-assisted selection more than phenotypic selection. Now, just very briefly, I'll quickly talk about some work we found with the VRN gene, which is the subject of a nice poster by Lee Hickey and others, poster number 49. For the wheat story, we looked at 3 near-isogenic lines developed by Ben Travaskis at CSIRO in Canberra. He's got ones with different winter or spring alleles at either the A, B or D genome, and they're in a sun state background, which is a spring wheat background from Australia. And if we look at those, we've got our sun state wild type with winter A, spring B and D, and we've got winter A and D and spring B there, and winter for all 3 in these particular nils. And you can see that it's the nil with the winter type that's got the narrower root angle that we're generally after. But if you put those plants then in these tubes and grow them through to maturity, if you look at them at anthesis and look at their total root biomass at different depths, at anthesis there's not a lot of difference between those genotypes. But once you take them through to anthesis, you can see that the winter wheat hasn't changed much from anthesis, but both of the spring types have grown more roots between anthesis and maturity. So this is a trait that we're after. So the spring allele there is favourable for that deep root proliferation, but actually the winter allele is favourable for the narrow root angle. So that leaves us with a quandary. What's the best allele combination that we can have for our environment? Do we need to be looking for different VRN1 alleles than what we've actually tried? And so that work was done by Lee Hickey, Hannah Robinson and Kai Vosfels, who's the main author of the paper, and Rod Snowden in the UK. So basically I outlined how our target environment has dictated which traits we needed to select for, showed you the nice population that Lee and Cecile developed for incorporating those traits into relevant backgrounds for our environments. We talked about that high throughput screening method that Cecile was able to successfully use to shift the phenotype for seminal root angle. And just a quick plug for the VRN1 work and the poster, and any questions about that would be, I'm happy to answer or refer you to Lee later on. So thank you very much for that.